Okay, so now the recording is on. Hold on, let me put the right box on so I know if you chat to me. Yeah. So, just to make sure, I know you're like, oh, this is the schedule. I'm horrible with dates. If you ask me, hey, November 8th, I'd be like, I don't know. It's just horrible. So we are in that week there. Right, we got 2 weeks. We're going to do chapter 11 today, actually. No, wait, we did 10. Mitosis. We did mitosis, right? I don't have any pretty cold clothes. I have just cold clothes. See? Good. We do meiosis. Then I thought we did. That's all. Okay, so we got the next 2 weeks. Next week, we're a little bit ahead, which is good. We did our lab practical last week, so I scored those. I didn't give them back, though. I will. 150 was the top, is the max, okay? So when you look and go, oh, I got 100, go, yeah, wait a minute. Anyway, I do have the key in my office. We can go over it. I have the questions. I've set these things up a million times. I can recreate the exact question if you want to look at it. Go, I don't get it. I'll see if I got it wrong. You guys did impress me on one thing, but I'll talk about that in class if I don't forget. So take this time to get caught up on your assignments, and don't take those things lightly, okay? They're 20 points and 16 points. Those things add up. Yes, Penny, you are. Yes. Yes, Penny, are you? No, hold on 1 second, folks. There we go. I have a new dog. She's a little bit overwhelmed. She's a rescue, older female. She's a snorer, quite a snorer. She's a little bit overweight. So if you hear that in the background, it becomes problematic, or like you can't hear me, then let me know, and I'll ask Emily to quiet down, please. That's her name, Emily. Actually, Bella. There's her whatever thingamabob name. Registered name, but she goes by Emily. All right. I love Calvin and Hobbes. This always reminds me of how, maybe this is why I raised my children the way I did. My kids always say, you raised us like Calvin and Hobbes. And I'm like, well, there was 2 of you, only one of him. But I always tell my kids I raised you both equally bad, so sorry about that. But I used to tell them, especially when they ask me stuff and like, Daddy this, Daddy that, because with kids they want to ask you a million questions. Let me turn my headset on, or my thingamabob on. After a while, I just go, Daddy, how's electricity work? Magic. And they were good with that for a while. I think till I was 17. Okay, we're going to talk about sex today. Awesome, right? So if you're thinking, oh, great, I tuned in to watch Mr. Clark destroy his career, well, good luck with that. I didn't teach this long to do something dumb like that. But we are going to talk about sex. We're going to talk about the naughty bits, but not the naughty bits in that particular way, but naughty bits in terms of reproducing. And first, before we talk about the naughty bit way of doing it, let's talk about the more boring but very effective, by the way, very effective way, let me see, there we go, of asexual reproduction. Now in science, when we put the A in front of a word, or AN, we use AN if the word starts with a vowel. Like, for example, you wouldn't say aerobic, okay, just because that sounds weird. I guess, aerobic. You'd say anaerobic, right? When we talked about that. But if you take a word and put the A in front, it very often, a good guess is it's the opposite. So sexual reproduction, well, what is sexual reproduction? Think about it. What did I say the other day about a teenager? But it seems to be important to us enough to, holy cow, do everything around it, right? When it comes right down to it, sexual reproduction is just a mixing of cells. That's all. That's what biologists say. However you mix those cells together, internally, externally, however that happens, however that sperm and that egg and that male gamete and that female gamete come together, sexual reproduction. The mosses were doing it, right? When we said fertilization, that little sperm swims out through the droplets of water from the antheridia to the, come on, let's take a base. What's that thing called? 1,000 points if you spell it right. 1,000 bonus points. You guys would kill each other for that. 1,000 bonus points. If you can spell it right, what that sperm swims into and fertilizes that egg, it's at the bottom of that vase, and out comes that's sexual reproduction, right? However you do that. I don't see any answer there. Come on. Either I'm lagging or you guys are trying to figure out how to spell it. Get it as close as you can. I told you, the antheridia, the sperm, and it swims into the... I'm not going to go on until someone says something. Unless I'm just sitting here. Is my internet on? Yes, it is. I always worry that I'm talking into nothing. Hello, Emily, Courtney. I'm going to start calling Penny. Yeah? Cool. This thing. The thing that the sperm swims into in the mosses. I'm talking about sexual reproduction as being really nothing more than sperm and egg coming together. So I said way back in the mosses, right? The sperm came out of the antheridia through the water droplets. That's why mosses grow where they do. Down to the egg in that vase-like thing. What's the name of that thing? In the mosses. When we looked at them in lab. I said the antheridia produces the sperm swimming into the moss. Holy cow, we looked at them in lab. In the fern, they were a little more difficult to see. They looked like a vase. But in the fern, they looked like a vase, like you were looking straight into it. But in the mosses, they were awesome because you could see it from the side. And some of you... Yeah, but the rest of... Come on, guys. If I say this word, and yes, you do. Oh, holy cow. No wonder you didn't. Oh, my goodness. That's in the next slide. I put these slides backwards the other day because I wanted to talk about this first. I'm sorry about that. Anyway, scratch that for a second. I'll get to that in just a moment, okay? Just a moment. Anyway, so you can do it asexually, and that's very simple. Like, for example, a bud, right? That is not two cells mixing together, right? We'll talk about that antheridium and archegonium. I'm sorry. The other day I went, wait a minute, too early to put that slide. Put it a little bit later, and then I forgot to remind myself. What's XD? But the thing with asexual reproduction, right, is the bud. That's not really that critter's name. The bud is exactly the same as the parent. That's a hydra. You might have saw them under a microscope. They're related to coral. That's what them little finger things are. They're stinging cells. Sometimes if you swim through the grasses, the sea grasses down in the Keys, and you're snorkeling, maybe you get this itchiness in your chest. There are little mini ones of these that are like, hey, stop it, and they're stinging you as you go by. It's not like a jellyfish, but they're related to them. Anyway. So, but the thing with asexual reproduction is the bud is exactly the same as the parent, right? Now, in biology and life in general, and I really talk a lot about more of this in bio too, is this idea that no matter how you do something, if you're still alive today, about 500 million years after serious life began on Earth, life began even earlier than that, 3.5 billion years ago. But if you're around today, it must work. So it must work in your environment, or must work in your environment at certain times of the year or the day, or certain times of the season. So being a clone of your parent is not really a good thing, right? Can anybody think of a reason why being exactly genetically identical, let's say a whole village of 100, and they're all genetically cloned, somehow they learned how to asexually reproduce? What's the bad thing about that? The good thing, it's real easy. Go on. Increase of genetic disorders? There you go, right? Whatever weakness. Now, organisms, we didn't talk a lot about evolution, but organisms have an advantage in evolution. And evolutionary biologists relate that back to sex, folks. If you have an advantage, it means you reproduce more than other organisms. But the problem is if everybody's the same, in other words, there's no better, no worse for a particular thing. Let's say the bubonic plague. The bubonic plague killed a third of Europe, 30%. In some cases, entire villages. But in some cases, entire villages except for this one house or this couple of people that may have been related, for example, too. And those people were just naturally immune. Like, not all of us got the measles, right? I don't even know if kids get the measles anymore, but when I was a kid growing up, parents would actually mix kids to give them the measles before school started because they didn't want the kid to be sick during school. So it was this horribly barbaric thing where they would take us over and play with the sick kid
But some of us never got the measles, even though we were exposed. We were naturally immune, all right? Now, asexual reproduction, what's the advantage? Fast, quick, bud, bloop, we're ready to go, right? Disadvantage, there's no difference. But if you're living in an environment that says, hey, it's cool if everybody's the same because this environment is very stable and doesn't change, and there's really no better and worse for this particular set of traits, then by all means do that. Hydras do it. These things up above, which is a really cool picture of the, I'm not even going to try my pencil, it annoyed the crud out of me yesterday. This is 2 bacterial cells. Well, getting ready to be 2. It's not 2 right now. But this is binary fission, remember? Now, don't mess me up. I know we talked about that. I'm sorry if you hear Bella, or excuse me, Emily. Emily. Goodness gracious. Isn't that cool, the chromosome? So we're just, the chromosome is finished and the cell's getting ready to divide. Isn't that neat? Now, that's a real cell. They obviously aren't in that color. Wouldn't it be cool if you could look under the microscope at school and go, whoa, it's been color enhanced, but nonetheless, that's neat. What about redwoods, though? Why redwoods? I don't know, because it's a tree, right? What about trees and animals and pretty much what about the rest of us, all right? What about the rest of us that do this thing called sexual reproduction and either don't or mostly don't engage in asexual? Because even though we don't. And most mammals Trying to think. I think most mammals don't. Some might, hold on. But in some critters, they can do both. But anyway, we're going to talk about those that do it sexually, all right? So we start down the bottom with mom and dad, and obviously mom and dad are different. Now, the important thing about understanding mom and dad are different is not that they have different genitalia. Believe it or not, in biology terms, is one of those things where you say, that's correct. There are some sharks that are that way, too. It's a very interesting thing, right? Very interesting thing. In some species, males are not important. The only thing that females need males for is difference. And that's the subject of what we're talking about today. You remember in mitosis last time? I know we talked about that, right? What was different about the cells? After the cell divided, those 2 cells, what was different about them? Or what was the same? Compare and contrast those 2 cells in mitosis. Mitosis. So they were genetically identical then, right? Is that what you're saying? Yeah. They were exactly the same. Now, would that work? Well, sure. It might work at that nice, safe environment. Is that how an organism like the Hydra asexually reproduces? Well, not one cell, but many cells, right? But regardless of how you do it, asexual reproduction, mitosis, produces many cells, but they're the same. Might be okay. Meiosis is very different. Meiosis is division. You're going to talk about prophase again and metaphase, but they're going to have numbers. And that's one thing I want you to put in your notes and put in your head right now so that you don't confuse these 2 processes. The mitosis one, right, from last week, it's easy to confuse them because the names are the same and what happens to protect the innocent. Yeah, if you haven't seen old TV, you don't know what I'm talking about. If I say a number after the stage, prophase 1, metaphase 2, if I say a number, 1 or 2, I'm talking about this stuff today, meiosis, right? If I don't say a number, is that what it was? If I don't say a number, Penny, I'm sorry, I'm going to look quick. Can you type in there what you need me to repeat? If I don't say a number, I'm talking about mitosis. If I say prophase, mitosis. Telophase, mitosis. Telophase 1, meiosis. As soon as there's a number, okay? So you won't, because in the exam I may ask you questions so that you know the difference between mitosis and meiosis. Very simple, go, all right, first thing I'm going to look for is that number. If he's going, what happens in prophase versus prophase 2, I'm like, oh, he's doing a mitosis, meiosis thing. He's got that number thing, right? All right, makes sense. Now, let's talk about the difference with meiosis. Penny, I'm sorry, did I repeat what you needed to hear? Are we good? Well, I was saying in terms of when I refer to the stages, like prophase and metaphase, if I put a number, I'm going to be talking about meiosis. If there's no number there, you'll see in a second because I'm going to go into the stages and you'll recognize the names again from mitosis. Okay, now, in terms of biology, meiosis is a very, very different kind of thing that it goes through. It is cell division. It has to go through the same process, but the result is dramatically different. Right away, when you look at this chart here, in the ovary of the female, the testis of the male, or the testes of the male, when they undergo this process that we're going to describe, what's really different about the cells they make? I mean, what stands out right away that's different? I mean, yeah, one looks like a sperm, I get that, but what's really different about the cell? The sizes, that's true. The egg cell is usually bigger because many times the egg provides a bit of a starter pack for the developing, has more nutrients for the developing fetus, right? I always say eggs are expensive, sperm are cheap. What else is really different about those things? I mean, really different about cells. Think about numbers, right? Think about what makes us each different. Think about what that means in that parentheses down below under the animals. Well, they are under the people. 2n equals 46, right? Well, think about what that number means. How many chromosomes do we have? You don't know what 2, yeah, right. Well, I'll talk about the 2n in a second. Now, that makes us all different, right? Not humans. Humans have 46. That's how we define species. One of the ways we do is by the number of chromosomes. Okay, that's what the weird thing about dogs and wolves are, because dogs and wolves don't have exactly the same number of chromosomes, or they don't have the same number of chromosomes. But anyway, don't want to get too far off on that. But look what happens during the process of meiosis. We make sure that we make a cell that keeps that number the same. I mean, think about it otherwise. If I made sperm that had 46 chromosomes and the females made with another 46, you can do the math, right? That's like 92 chromosomes. Not good. Not good to have. Not good. We're going to learn in genetics. Just sometimes not good enough to have an extra one, let alone a whole. That kind of stuff does happen too, by the way. In very interesting twists, but unfortunate for the folks, or for the humans in this case, right, that suffer from it. So we make these cells that are not the same number of chromosomes, they're half the number of chromosomes. So that's where the name diploid, haploid comes from. Get it? Haploid are the cells, egg, sperm. We call these things gametes. These cells collectively, egg and sperm, are called gametes. These things have half the number of chromosomes. And then, of course, just do the math. When the sperm and the egg come back together, you add the 23 in the egg, 23 in the sperm, and boop, got yourself junior. Let's call that a zygote. See, fertilization, a zygote, a diploid zygote. Back to that number. And then guess what happens? Then it's simple. You just start cranking on cells. Boom, 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 making them different, making them different. See, the cells are going to differentiate, remember? They're going to become liver cells and kidney cells and heart cells and all the kind of different kind of cells. But you're going to make a lot of them from the same base. You're just going to change what genes turn on. You're not going to change the genes. Does that make sense? Diploid, haploid, diploid, haploid. So if I talk about those, you don't confuse them. Just think haploid half. Diploid, di, like a disaccharide has two. Make sense? Everybody cool? Okay, sit back and rest for just a second because I'm not going to test you on this, but I did want to show you that there is another way to do it that I find kind of interesting, and this is where I was confusing you guys just a second ago. In many species of plants, like this fern that you see in the picture, they have something called alternation of generation. And what that means is that it alternates, obviously, between two generations. But this thing down below that is haploid, how do I know? Because it says so right there. This little thing is called the gametophyte, all right? And in some species, like this fern, they actually produce multicellular haploid things, multicellular haploid structures. And the reason why they do that, it's simple, folks. If you know that most of your sperm and egg, if you know most of the sperm are not going to make it down here into this thing that looks like a vase, there we go. This thing is called an archegonium. Don't worry about the word, I don't care. The other thing is called an antheridia. I just shown you something different. There we get fertilization back into a zygote, right? We're back up into the diploid. But these guys are going with numbers. Make a lot of them. Why so many? I mean, we aren't up to our butts in ferns, are we? Thank goodness. It wouldn't be so bad. Ferns don't bother me. Why make so many sperm and egg? I mean, humans don't make, we make a lot of sperm, but there are some critters, and then even after they fertilize, they produce tons and tons, and yeah, how does it? Come on. How does it ensure survival? Survival of what? Not the individual. I mean, if you make sperm that are messed up somehow, there you go, right? Survival of the species. We're back to that whole idea. Yeah, only a few. Yeah, it's right. It's all about the idea that if you don't know what the environment's going to be like, and nobody does, we can't predict the future. Organisms, we sometimes forget as humans about evolution, about how it works. Organisms are trying to figure out, well, I'm going to make as much difference as I can. This whole chapter, folks, is about variation. That's so important for understanding, I mean, from a biological, from a sociological point of view. It's understanding the roots of what makes things different, okay? It's important to understand how this came about. That's what we're talking about. So when we look at organisms who do it differently, they make sperm and egg, but they make sperm and egg producing factories. They're going with numbers, right? Now, as we move along the way, if you think about moving along the way towards us, we've changed, haven't we? We don't go with numbers. We go with fewer offspring, but more parental care. It's a balance. Everybody along the way sometimes finds a niche. Sometimes somebody messes up and they become extinct. The dinosaurs, right? Reptiles didn't become extinct. Dinosaurs really didn't become extinct. Can you believe that? Dinosaurs did not become extinct. The birds, we've now decided birds are really just dinosaurs. I love that. You got it, and there's a formula to calculate that, Seth. Can you believe it? It's called Hamilton's rule. Don't need to worry about looking at it. But it's an interesting formula that says if you're standing there on the beach and your cousin's child is drowning, that evolutionarily there is a calculation that occurs about whether you should go save them. What's the question I used to ask in my biology class? My bio teacher used to say, is it better to save your son or 4 cousins? And if you do it with Hamilton's rule, there actually is a number that says it's better to save 4 cousins, I believe, if I remember correctly. Hey, what's this? I wish, man, they had these when I was in school. We used to make these. They used to give us a photograph of a bunch of chromosomes. These chromosomes are condensed, right? That's why they're thicker. We talked about condensation and mitosis. So there's a lot of similarities that's going to occur in these two things. So be very careful. Remember, if it's got a number, I'm talking about this one here. But these are chromosomes, and the reason why they're paired up the way they did, and we talked a little bit about this in mitosis, is although we have 46 chromosomes, we got 23 from Ma. Those we would call our maternal chromosomes, and 23 from Pa. Those we call our paternal chromosomes. So the one that's got the box around it, we might separate that and say there's a number 1 maternal and a number 1 paternal, right? One you got from your mom, one you got from your dad. We really have 23 pairs of chromosomes, don't we? We have 23 pairs. We say they're pairs. Now, there's another word for the pairs. We call these pairs homo. They're the same. Logos. L-O-C-U-S. Locus. Without the T, not like the insect. Means a location on the chromosome. The little banding colors that you see there, see, when I was in school, they didn't do it. They did it in black and white. No wonder I can't see now. But the banding colors are there to indicate different regions of the chromosome. So homologous, what word is he saying, means same regions. All right, now, what we mean by that is the information, a gene that's on the sperm, there's also a gene on the egg. Same gene. Say that gene helps, or that gene controls hair color. It's not quite that simple, but you get the idea, right? Now, those are both the hair color genes. Now, when they come together, when fertilization occurs, then we go that old thing you might remember from doing genetics problems. Which one's dominant? Which one's recessive? Which one's this? Which one's that? If that makes sense, great. If not, don't worry about it. We'll talk about that. But get the idea that when those two cells come together, they're bringing the same gene, hair color gene, brown, blonde, and then that all gets worked out genetically, okay? Make sense? They're the same regions, but they're same genes, but they might have different information. Cool? This is called a karyotype. Kary, you'll remember, is a nucleus. What's in the nucleus? The DNA. This is the type of the nucleus. These are the chromosomes. Isn't that cool? These are human, by the way. How do I know they're human? I can see that there are 23 pairs. There's actually numbered for us. They are numbered too. Number 1 is long. You notice like the ones that were a pain in the butt were the number 19s, because they'd be black and white in different shades. Even though you say, what, 2 different colors? I mean, the other ones were all a pain, and they didn't line up. Sometimes they were curled, and you had to look at them. And this is all in black and white. Thank you. My college career has caused me to go blind early. Now they're all laid up, right? You got a number 1 through 22. This is obviously human, right? 46 chromosomes. We call the first pairs. I'll add to that in just a second. Autosomes. Okay. In our case, in humans' case, we have 22 pairs, 44, 22 pairs of autosomes. That's because the last pair, Emily. I don't know if you guys can hear, but my goodness, the dog can snore. Down here, oh, come on, Pam. This one down here in the lower right, take a look at those. They don't have a number, do they? They have a letter. Sorry. Emily. Come on, baby. Just kind of roll. Oh, there you go. You can't nap all the time. I love it. Male or female? Anybody know? Is this a karyotype of a male or a female? I know you guys know this. No. No, remember who determines the sex of a child, right? This is a male, XY. Females are XX. We'll talk a little more about that when we get into genetics. But anyway, that last pair. So you don't always have 22 autosomes. We do, right? But if you had 10, 8 would be autosomes. The last pair are called the sex chromosomes. They're called the sex chromosomes for quite obvious reasons, right? XY, you're a male. XX, you're a female. They both have different sets of genetic information, not all of which, by the way, interestingly enough, has to do with sex. Not all of which has to do with your sexual characteristics, the genitalia you have, the cells you make. We'll talk more about that later on. I might explain if you're thinking, I wonder why boys always seem to be higher rates of color blindness. Really? Yes, they can. To their boys, they have to. And I always say colorblind daughters also have to have colorblind fathers, but I don't want to go too far on that right now. We will talk about that. It's very cool. I love it. It's neat stuff. Okay, so we know what a karyotype is. We know what autosomes are. We know what sex chromosomes are. Pair sex chromosomes. All right. Let's kind of take a real simple cell. First of all, let's review this whole idea of what the heck is a chromosome and what's a chromatid. Let me see if you can answer this for me first. In this cell, how many chromosomes do you see? I'm going to step away for a second. Next, I'm going to want to know how many chromatids, and I'm going to want to know how many pairs of homologous chromosomes. All right, so very cool. Penny's got it. We got 6 chromosomes. That's excellent, right? Got 12 chromatids. There we go, right? Remember the idea, and you have to be careful about this because it's easy to get confused because it starts out chromosome. What's the difference between a chromosome and a chromatid? Really nothing in terms of structure. It's only the copies of each other, right? That's why we call them, look up here, we call them sister chromatids. You're only a chromatid, okay, when you have your copy attached to you. Otherwise, you're just a chromosome. They're the single copies there, Jordan. If we looked at this and this cell had not replicated its chromosomes, quite clearly they have. You see the copies are on each one, right? We would see one long blue one, right? One medium blue one, one short blue one, and then the same thing for each of the red ones too. We still see 6 chromosomes. Chromatids only exist when when chromosomes are replicating. Think about chromosomes as being really sort of set of information. And since the chromosomes have replicated, there's nothing different between them. They have the same information. We're all clear? Everybody knows that the centromere, that's the middle part. That's where the spindle fibers are going to attach. That's going to become important a little bit later on, but we'll get back to that. Look at what we're calling a homologous chromosomes, right? Down there on the right. There's a pair. All right. It's not. Maybe I'm overthinking it, but I remember when I was going through this in school, I was getting very confused because I'm very bad at visualizing numbers and this kind of stuff. But notice why they're calling that a pair of homologous chromosomes. Okay? That's 4 chromatids, but it's a pair and of homologous. There's the male, that's the blue one, let's say. The red one is the female. Now, also notice why they're calling those non-sister chromatids in that pair. Because they're not the same copy. The copy of the red one is the copy of the red one. The copy of the blue one is the copy of the blue one. But then the ones up above, those are sister chromatids, right? And there's the word maternal and paternal again, and there's the number, right? We're just kind of setting the terminology here because I want to run into the stages now, and I want to make sure we're all very clear about when I say something like homologous chromosomes and sister chromatids and all this kind of stuff, right? It can get a little bit confusing. Cool? The stages of meiosis, all right, are the same in terms of the names as I said before with mitosis. There's going to be an interphase. Matter of fact, we'll start off very simple. There's only one interphase. It's not interphase 1. It's just called interphase. And what goes on in interphase, if you want in your notes to make it nice and simple, you could actually go and just say C mitosis. All that stuff is happening. G1, S, G2, these cells are going through the cell cycle just like all cells. These are actually special cells. I mean, all of your cells are doing that because they're doing mitosis. But in cases of meiosis, so are these cells that we call gonal cells. And gonal cells, as you might imagine, are produced there. Gonads. I always kind of wanted to be we had flag football at UCF, and I always wanted our team to be the Nads. I'm stupid. Go Nads. Anyway, those cells down there are undergoing interphase. Okay, they can also undergo mitosis, by the way. Gonal cells can also divide as well, but we already know how that works. I want to talk about when they go through the other process, when they go through this really cool process, this thing called meiosis, right? Because they're going to do a couple of things, right? Number one, you already know. They're going to divide the number of chromosomes in half. We're going to see how that happens, but it has to happen because sperm and egg, right? They're only haploid. They're not diploid till they come back together. Number one. Number two, does anybody know what the second reason why meiosis is going to occur? It's not for quantity of cells. It's to divide the number of chromosomes in half. But the reason why I'm asking, you might think, well, we haven't talked about it yet. Think about it in general. Is everybody the same? Even if you have brothers and sisters, same parents, right? Are y'all the same? I mean, the same. Everybody's different, aren't they? Even in your family, sometimes you look and you say, holy cow, where did the red hair come with this kid? Oh, my great-grandma, or whatever, right? We're all a little different. And we talked about variability before. Y'all understand why that's important. But you notice that what also must happen is that means if we're all different, all of our sperms are different. All of our eggs are different. Different. So what we're going to discuss in meiosis is not just some simple math thing of a diploid cell of 6 chromosomes. Yeah, buddy, it is, Jordan. Sorry, I didn't mean buddy. We're not just dividing a cell of 6 chromosomes into 3. That's the simple part. That makes perfect sense. We're talking about making everybody different. Now, just to really blow your mind, I'll give you the number early, but the chances of you producing the same offspring, okay? So if you think, in other words, you have a kid and it has a genetic makeup. 5 years later, you have another kid and he's exactly the same, down to sex, down to everything. Is that possible? Well, sure it is. There are identical twins, but I'm not talking about that. I'm talking about the fact that it's the same parents producing the same sperm, the same egg. It's all very random, right? It's all very random. When you win the lotto, it's not because the numbers, my wife says, oh, 3 of the numbers came to me last night in my sleep. I'm like, what does that win me? You know, give me the other 3, maybe we'll play them. But I did the calculations. I don't remember the number, so I'm going to have to do it again, folks. I'm sorry. I was doing it this morning. It's one of those big numbers that I can't remember. And I'll show you the number again later on. But the odds of you producing the same kid, in other words, the male makes the same sperm through this random process we're going to go through. Think about rolling the dice, right? Let me do that right. You're going to love this. I don't even know what this number is. Let me see if I can try it. Oh, gosh. 70 trillion. Yep. You have a 1 in 70 trillion chance of the male in a mated pair producing exactly the same kind of sperm through this really random process, the same in the female, and producing. You have a chance. You have that chance. 70 trillion. There's not been that many people alive since Earth has been around. I think there's only been, I don't know, maybe a trillion or something. What comes after a billion? Million, billion? Maybe 100 billion. There's no way there's been 70 trillion around. So let's see how this happens. This is very cool. So interphase, blah, 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 right? Not blah, 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 but make sure you understand what's happening here. Let's do a numbers game. Start there at the top cell. Very simple. How many chromosomes do you see in that top cell?
Emily. Why don't you move a little bit?
Why don't you pick me up and move me? Yep. I see a maternal and I say a paternal. All right, just for fun. How many chromatids do you see in the top cell? I don't see any either. There ain't no duplicating happening, right? Let's follow down to the next cell. Same thing. Do you still see 2 chromosomes? And do you now see 4 chromatids? Excellent. All right, now let's remember that number. I'm going to write it down. Let's see if my pen works. Probably not. So we're going to start up here with a cell that is obviously diploid. How do I know it's diploid? Well, it's got a paternal and a maternal. This could be the number 1, the number 17. Thank goodness they didn't try to draw out all 23 pairs, right? What a mess to try to look at. It's going to happen the same in all pairs of homologous chromosomes. So we're up here at 2n. Oh, good. Thank you. You're working. I'll go with numbers with humans just to kind of, give you a basis here. So when we get down here to the next cell, we're still 2n, and my pen decides to stop working. Right? We're still 2n. We're still 46 chromosomes. We might be 92 chromatids, right? But we're still that. Let's see what happens in meiosis 1. There we go with the numbers. So there's no interphase 1, but there is a prophase 1, which a lot of stuff goes on, so we have to talk about that, but it's absolutely incredible. Metaphase 1, that's an easy one to recognize because it's right there in the middle, right in the middle. Anaphase 1. There's a telophase 1. There's cytokinesis. The cell has to divide, and that's going to be different whether we're talking about this stuff happening in an animal or a plant, right? Y'all remember how animals undergo cytokinesis? Remember the name of that process? I'll save you guys time flipping through your notes. And then, of course, in plants, it does it this way, right? Because they have a cell wall, it makes it a little bit different. So You know, in terms of the mitosis and meiosis, there's a lot of similarities. That's why it's very important that you be very careful that you don't confuse them. And I've got a couple slides try to help you do that. All right, let's go back to the numbers game. We've got 2 cells now. So we got meiosis 1. Prophase 1 has happened. Metaphase 1 has happened. Anaphase 1 has happened. Telophase 1 has happened. Clearly, right? Cytokinesis has happened. We're at the end of telophase 1. There is no interphase 2, so we're going to go right into prophase 2, but we're going to stop right here for a minute and check our numbers. How many chromosomes in each of those cells that came out of meiosis 1? How many chromosomes do you see in each of those cells? Chromosomes. Not human. That would be correct, by the way. Just in this one here. Chromosomes. Somes. I see. Right? Chromosomes. How many chromosomes do we see? Here's where it might get a little bit tricky. Well, it looks like to me like they're connected. Each cell. How many chromosomes do you see in that cell? Right there on the left-hand side, the one with the blue X right there. I see 1. Does everybody see the one that Seth sees? I see 1 chromosome. But Penny, I do see 2 chromatids, right? I see 1 right there, and I see 1 right there. It's easy to do. That's why I'm emphasizing this. It is easy to do. Okay, so the cell on the right, well, we still have 1 chromosome. Now, think about what we need to do in meiosis. 2 things. The simple part, the math. We need to go diploid to haploid. That's why I said Courtney's answer was correct if we were talking about human beings. All right, I'm sorry, Courtney, you might have got mistaken since I was using human beings as an example. Now, look at the name of meiosis 1. Isn't that neat? Reduction division. What happens in meiosis 1? We produce cells. No. I haven't gotten there yet at all. I'm still right. I still have to go and finish up meiosis 1. Right? But see what happens in meiosis 1? Now that variability part that we're going to talk about, that's going on too. But we're kind of wait to talk about that to first get this numbers things figured out, right? So after meiosis 1, after prophase 1 and metaphase 1, after telophase 1, after cytokinesis occurs, and bloop, they get into 2 cells, we have accomplished half, at least when it comes to of our overall goal here, right? We have made them variable, but as far as the numbers go, we've accomplished that task. We've reduced the number of chromosomes in half. We've taken whatever you got as a diploid number, I think, like, for example, tobacco plants, I don't know why, 78, and we've divided that into cells. What, isn't that 39? Whatever that is. Gosh, I'm horrible at that, right? We've divided the number of chromosomes out. Now, we're not in meiosis 2 yet, Jordan. We're stopped right. We're stopped right here. Everybody cool with me? Everybody up to speed on what we're doing there? Excellent. All right, now, down below, we're going to go through prophase 2, metaphase 2, anaphase 2, telophase 2, then cytokinesis again. So imagine a little bit further down the road, these cells have separated. Man, I wish they would have drawn that since they stuck with that. These are separated, okay? So we get 4 cells. 4, because the blue one up above, it divides, and the red one divides. So we get 4 cells. So out of meiosis, because in meiosis you have to go through both stages, out of meiosis, what we end up with is 4, okay, haploid cells. 4 haploid cells. Everybody follow me there? As a matter of fact, meiosis 2 is sometimes called, which is a little bit confusing, it's sometimes called mitotic division, because if you look at those each by themselves, like just the red one, doesn't that look like what we talked about in chapter 10? The X's split, right? The spindle fibers pull them apart here and here, right? Same thing's happening over here too, right? So that's kind of like mitosis. What I don't like about it is the confusion part, but Is anybody confused about this whole idea of what we're doing with the number of chromosomes? How we're dividing them in half. At the end, you're going to get 4 cells. Each one of those is haploid. Make sense? I'll ask a question before we leave, so you got to think about this. Okay. So let's imagine they're divided. You got it. How many chromosomes do I see in each one of those 4 cells? In this cell right here, how many chromosomes? How many there? Keep going, come on. Somebody else. Who's in the room? There we go. Every single one, right? How many chromatids do you see? Penny, there's 1 in each one, right? I don't see any chromatids at all. Why? We don't have this. We don't have a duplication, right? That's why there's no chromatids. Whew. We good? Let's tear these things apart. This is so neat. I love because I love talking about it, like snake bio too. I just love talking about variation, how it happens. All right, so to remind you what's going on, down below in the red or in the dark red, whatever you want to call it, I remind you that meiosis 1, the purpose of all them stages with a 1, prophase 1, I'm not going to talk about anaphase, it's all the same. Metaphase 1, anaphase 1, telophase 1, and then, of course, this is obviously an animal cell, right, since it's undergoing it by cleavage furrowing. The purpose of all of this, overall, mathematically, is to produce these haploid cells. See, after that, all of these cells are split into 2, right? All of these cells, or these 2 lines of cells, they're already haploid. That happens right there. You got to remember chromosome and chromatid. Now, the picture here, the chromosomes and the chromatids are a little bit small, so I don't think I really want to use this picture to illustrate. I'm going to split these in half, and we'll take a look at the first stage in a little more clearly. At the very end, this number 2, what we produce are 4 cells, right? Prophase 2, metaphase 2, anaphase 2, the 2 that were there, they split again. We produce 4 cells, but the important part, which is not illustrated right here, but I'm going to, is they're different. They're genetically non-identical. That's where the word daughter actually means. Daughter refers to the, I don't know how sisters become daughters, I'm sorry, but daughter refers to things that are from the same line but genetically different, which I guess daughters really are, so that's the name. Let's look at the first set of stages first. There's something that's going on during those stages that you really need to know about. Prophase 1, metaphase 1, anaphase 1, telophase 1, right? When you look at the picture, maybe I can make it a little bit bigger. Let me try that. When you look at the picture in prophase 1, that's not so bad. Let me try just a little bit. You're probably thinking, oh my God, is this guy blind? Yeah, basically. Oh, that's good. That's good. All right, so we got centrosomes, we've got centriole pairs, and we got spindle fibers. Again, if you want to just make it easy on yourself, you could say, you know, centrosomes, etc. Same as mitosis, because those things, this is obviously an animal cell. I hope that's apparent to you, except for the fact that the spindle fibers, they occur in both. But the centrosomes and the centriole pairs, those little, you know, perpendicular shaped cylinders there, those are indicative of being an animal cell. Does anybody remember what these things, these radiating lines, what, come on, man. Anybody remember what those radiating lines are called? Well, they are made of those, yeah, that's exactly right. That's what they are, they're micro. Does anybody remember what those ones that radiate out? Yep, it's asters, stars, think stars, asteroid, astronomy, right? And then Courtney said these are microtubules. Do you see if I mentioned to you that in that picture, so I'm looking at that middle cell right there, if I said, you know what, I can see both kinetochore microtubules and I can see polar microtubules. Would you guys be comfortable understanding what that is? Would you say, oh, I understand what he means. I understand why he's referring to them as 2 different kinds. Okay, so you notice that some of the microtubules are attached to the centromere. See the one right there? I'm trying to draw with my pen, excuse me. Right there. See how it's attached? And the one on the other side is attached to it as well. They're attached to a protein on the centromere that's called a kinetochore. There it is right there. That's the word. You got it. But if you look right next to it, as Courtney says, the ones that are called polar, because they go from the poles, they go all the way from the top to the bottom, see? They're not connected to a kinetochore. They're overlapped. Remember, they're overlapped, but they aren't connected to anything, nor are they connected to each other. That's important, right? Because one of these groups is going to pull those chromosomes apart because it causes them to travel. The other one's going to elongate the cell so it gets ready to pinch in the cytokinesis, right? All right, let me get my scribbling off of there. Let's go back to stay in prophase 1, right? The stages run pretty quick through there. Another thing that happens in prophase 1, very different than what happens in mitosis, and back to this whole idea of what we're doing making variable cells, is you'll notice that the homologous chromosomes line up with one another. They actually, it's like a jigsaw puzzle. Because of their chemical makeup, they know who their homolog is. No. You let me use that word. The number 2 paternal knows to line up with the number 2 maternal. Do mistakes happen? Yeah. Yeah, sometimes. Yeah, that's right, Courtney. Thank you very much. Nonpolar microtubules. No, they're not connected. They're not connected. They overlap one another, but they're not connected. They're not connected to the chromosomes. That's right. I'm sorry. This pairing up, so yes, there are mistakes. Sometimes the number 2 paternal ends up with the number 7 maternal. Mistakes happen, right? But this pairing of homologous chromosomes, we say, is a state. It's called a state of synapsis, right? It's synapsis, S-Y-N, means together. So they're laying next to one another. Now, there's a reason why they're laying next to one another, and we'll get to that in just one moment. They don't really lay like this, though. They actually, if I had school, I could do it. But if I had 4 magic markers and I made them into a square, I could draw it, couldn't I? So here, remember, I'm right-handed, although I'm left-handed. So they're like this, really. You guys dare play the chromosome chromatid game with this thing with me? Oh, we're going to anyway. How many chromosomes do you see in this cell? If they can please, how many times we're going to do it until I feel like y'all know it. How many chromosomes? Yep, I do too. Does anybody not see what Courtney sees? Okay. How many pairs of homologous chromosomes do you see? I do too. Now, for 1,000 bonus points, you guys obviously know that's not true. How many chromatids do you see? That's right, Penny. I do too. Y'all? Anybody not see that? Everybody cool with that? Make sure you let me know. It takes a little bit of time to get familiar. There's a lot of words to know. Gosh, when I learned advanced genetics about mitosis and meiosis, there was dozens of substages. I think there's 5 substages of prophase 1 actually. So back to my question that I posed a couple of minutes ago. Why are they in synapsis? Why are they laying next to one another? Well, I'm going to have to go to a blank whiteboard, and I'm going to have to draw this out, and if hopefully everything's the way it's supposed to be, my pen is working. Let's hope it is. Thank goodness. All right. So I'm going to draw this out. And I'm going to draw out a pair of synapsed chromosomes. So they're connected. I'm not going to get too crazy because I don't want to make too much complication out of this. And so these are the, I don't know, pick a number. The number 7 paternal, and it rightly found, again, they're like jigsaw puzzles, so they fit in, like a substrate and an enzyme, right? So they rightly found its chromosome, and it's laying not next to one another, but again, kind of like on top of one another. So they're in synapsis, and they form, with those 4 circles that I drew in the previous slide, they form something that has the word 4 in it, doesn't it? They form something called a tetrad. So if you take those 4 things and stack 2 on top of 2, they look like a tetrad. But I'm drawing this way because I need to for a reason. Now, excuse me, one of the first things I want you to notice is the fact that there are spindle fibers going this way. Or not notice, but I want you to know. There's one there, and there's one there. But there's not any in the middle. Okay, now that's going to have serious implications here in just a moment. But I want you to notice that first. There's no spindle fibers that's between those pairs of homologous chromosomes, but there is one that's pulling it this way. And if you remember from mitosis, it's the cohesion proteins that stops them from separating. So you might be thinking, well, what's in between here? How come they're not pulling apart? There's no glue, there's no cohesion proteins, and I'm really sorry for that horrible drawing here. Anyway, what is keeping them apart is something, I'm not going to write it, I'm going to spell it. It's called a chiasma. I always have to refresh myself which one's plural. Chiasma. Chiasma or chiasmata, which are plural. PL, that means plural, right? Chiasma or chiasmata, right? What chiasma or chiasmata are, what it literally means is, remember CH in science is not ch, it's not a chiasma, it's a k, like in chitin. Talked about chitin. I'm going to have to erase the board, I'm sorry, it's just kind of the way, it's a weird way that this works, but I'm going to come back and recreate these. Here's what a chiasma looks like. Let me get my pen again. I'm going to draw this chromatid like you see before. And I'm just going to draw one chiasma. Now, in a human cell, on average, when these homologous chromosomes get together, on average, there are 1 to 3 chiasmata that form along these chromosomes. Let me show you what a chiasmata is. And by the way, those 1 to 3 that form, random, random, random. Sorry about that. They're all, I shouldn't try to talk and draw at the same time. It's random where they're formed. All right, so there's that one there. This one here I want to draw a little bit different. I'm going to draw it kind of like it leans out a little bit, all right? And, of course, there's that other pair, and it's also leaning out. So if you permit my really crude, it doesn't really touch that other chromatid, sorry. But they cross over. It's kind of abstract, but you get the idea, right? So that crossing over point is a chiasma. And on average, along those paternal chromosomes and maternal chromosomes, along those homologous pairs, those chromatids, and they can cross over like this one here could then go over and cross over with this one, right? So and this is all happening randomly. Where these chiasmata form along those 23 pairs, how many form is all very random. So what happens, and you're going to see how it happens in just a second, but what happens is you're going to make something that's called recombined DNA. You heard a lot. They use the word recombinant DNA, and you think, my goodness, what is that? DNA that's been mixed up. That's what that means. Exactly, Seth. That in sexually reproducing organisms, there are a variety of ways you get variation. In sexually reproducing organisms, right, this is the #1 way you get variation. Most of the variation that you see of genetic, of organisms on Earth, the differences that we see come about through this random process that occurs during prophase 1, this crossing over process. So if you'll let me kind of continue with this drawing, let's see if I can do it without really making a mess out of it. Let's talk about some genetic traits. Let's talk about some genetic traits that have 2 choices. Tongue rolling. Either you can or you can't, right? So let me write that down. I'm going to say tongue. Is it a G or a Q? Whatever, it's a G today. So if you're a tongue roller, you only got 2 choices. Either you can or you can't. I don't know the evolutionary significance of it, although I should look that up because it was kind of weird. We'd almost think it has something to do with nursing, but or those who can't roll their tongues is fine. Earlobe attachment, that's another good one. If you think about, well, all of our earlobes are attached. It's like this. If your ear has a lobe to it, yeah, that's hard to do. Then you have unattached earlobes. And if you're like me, my earlobe kind of goes straight in the side of my head, then you have attached earlobes. All right, so I wish I could erase part of this. Let's imagine that the genes for tongue rolling were contained up at the top of these chromatids. The locus, that's the location. LOCUS, widow's peak is another one. Yeah, you either have one or you don't, that's true. It's also something with your fingers, something like that. So let's let me get a different color actually. Let's try red. So let's say the gene for the locus for whether you can roll your tongue is up here at the top of the chromatids. So we'll say, all right, this area, and I'm only picking the tip for sake illustration. That's all, folks. That's all. It could be anywhere along that chromosome. So, of course, if that's a copy, then that's the same locus where the tongue rolling gene is going to be found as well. And, of course, they're homologous chromosomes. And that is not just a word. It's same genes. So the tongue rolling gene, and just, again, for sake of argument, I'm going to make it a different color, is on the same place on the other set of homologous pairs, right? It's up here at the top. Now, I turned to green only because it's different. We could say, for example, green is no, that you can't roll your tongue. We could say, for example, that red is yes, you can roll your tongue. Now, if you're thinking to me, if you're thinking about, oh, that's this dominance and recessive thing, did I draw one of those? Widow's peak. Oh, pull your hair back. You look like Eddie Munster, like the hair comes down in a V, or you don't have that. Whether you can roll your tongue, as I was saying, whether you can roll your tongue, yeah, is going to be decided by genetics. So don't worry about that right now. Well, let's take the other one. Let's take the earlobe one. And let's do the same thing. Let's kind of stick with colors. Let's say red, yes, you've got unattached earlobes. Green. Excuse me. Sorry, folks. Best plans of mice and men. Not this color. Green is this color. All right, so let's go ahead and draw those colors out again. Let me start with the red. So I'm going to say down here at the bottom, on this, there's, and again, I picked the bottom just because it's simple, that's all. But now, on the one that's crossed over, the bottom is now over here, right? And then I'll pick the green one. This one here is the same. But notice now that over here, that's the same. So in other words, wherever there's a chiasmata, the genetic information is moved from one non-sister chromatid to another. That's why it was important for you to know those terms. See, that chromatid that's crossed with the one in the middle doesn't have to cross with just the one in the middle. It, as the arrow, it could cross with this one over here. All right? Now, I'm going to have to let you visualize that, and I don't want to erase this drawing, but I want you to visualize what that's going to, well, no, I'm not going to erase it, but I'm going to continue drawing. Look, what you're going to end up with is 4 different chromosomes, aren't you? Once they're separated, they're not chromatids anymore. But look, you got one that's green on both ends. This is going to swap. All right, so you're going to have green and green, green and red. You're going to have red and green, and then these both are going to be red. Now, let's take a breath for just a second. What I want you to tell me is just based on the colors that are on those chromosomes, the chromatids right now, I understand, but when they separate, they'll be chromosomes. Are those 4 different? Are there any that have the same colors in the same orders? Not where you're sitting. Well, Penny, let's take a look at it. This is going to be chromosome number 1. Let me get a better color than this, right? Let me get a black. There we go. Chromosome number 1. Let's write this down. What's the genetic information? Now, the genetic information is indicated by color. Green, you can't roll your tongue. Green, you have an attached earlobe. No, don't do that right now, because that'll confuse everybody. Well, oh, green. I'm sorry, Seth. Beg your pardon. Yes. The number 1 chromosome there is green and green, right? The top is green, the bottom is green. What is the color order for the second soon-to-be chromosome? Green and red, right? How about the 3rd chromosome? How about the 4th chromosome? So we got green, 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 red, red, green, red, red. Are any of those the same? Not a single one. They're all different. Now imagine human beings have somewhere in the order of 30,000 genes. 30,000 genes, but when they get together, they randomly swap information. So think about that in variability, all right? Now this is all happening in synapses. This, what we call crossing over. That's what this is called because obviously. Crossing over, this is when it happens. In prophase 1, we have, if you're thinking, my God, it's going to take all day, don't worry, the other stages move quick. But this is in prophase 1. Synapsis, then they're going to cross over. All right, now, they haven't swapped their information yet. They're crossed over because they're still attached to one another. So now I'm going to go, where do I want to go? I need to go back to the slide, so give me 1 second, folks, because unfortunately it doesn't work quite as quickly as I would like it to, unless there's a way. And here, well, I'm not too far in, so that's good. All right, so we're all good here before I leave this picture. Anybody want to ask any questions about this picture before we go? This is what we would be seeing if we were looking at a cell or a pair of homologous chromosomes up close in prophase 1. And I've just picked locus, or loci is the plural of that, by the way, loci. I've got several loci and just colors, just at random, just to kind of show you how you could shuffle. And that's just, heck, that's just 2 genes, man. This is one. We're ready? Some give me a yes or a no, or give me a thumbs up, or hold on, or something before I go, because I don't want to recreate all this. We're all good? Look at the picture on the left. All right? You know what I don't like about that one on the left? Does it? No, it doesn't, does it? So you can do the same thing with the picture on the left. The paired of look where this slide is actually wanting to emphasize where the spindle fibers are. All right, notice where the spindle fibers are. See, the spindle fibers aren't in the middle, but if you take a look, we can do the same thing. So these are 4 chromatids, but they're going to be separate chromosomes. They're going to end up with 4 cells. So this is at the very beginning. This is metaphase 1. But let's do each one of those chromatids eventually going to be different kinds of chromosomes. So we got red, red. Then we have blue, red, blue. I'm going to write this down. We got red. Then we got blue, red, blue. That's different genetic information. Red, blue, red, whatever, and blue. 4 different ones, right? So same sort of idea as we did before, but those are going to be 4 different kinds of chromosomes, aren't they? And those chiasmata, now we're plural, now that's what's holding them together. Remember I said there's no cohesion proteins. Still chromatids after the mixes, they are different. Nah. There might be a place where they change, in the terms. Remember, these stages are flowing together, so it's not like the cell goes, oh, prophase is done, time to move on to metaphase. That's why, like I showed you in cytokinesis and telophase. You remember while telophase was settling down in the nucleus, the cell was already going into cytokinesis. So to answer your question, somewhere along there it probably changes. But we probably still call them chromatids while they're attached, just to keep it simple, Seth. But that's a good question. Okay, so it is the attachment thing. Did you look that up? Or does your book say that? And I should know that. I think you're right, though, Courtney. I think you're right. I think it has to do with the separation. But, again, focus on the spindle fibers. Just for comparison, look at where the spindle fibers were in metaphase of mitosis. Notice no number, right? Metaphase 1 on the left, meiosis. Metaphase on the right, mitosis. Don't worry about the text so much, but look at the spindle fibers. There isn't that in between. And remember, these spindle fibers are pulling. They're under tension. And the only thing that keeps these chromatids, from separating and becoming chromosomes is the fact that there's a cohesion protein there, and this one's pulling this way. But there isn't something like that here. It's the chiasmata that are holding it together here in metaphase 1. See, it's the chiasmata, the crossing over points, the looping is keeping that pair of homologous chromosomes together when they're under tension, okay? But eventually what's going to happen So let's go back here in the metaphase 1, is those chiasmata are going to break. And I don't remember exactly what causes that, but regardless, the break is going to occur, and those pairs of homologous chromosomes are going to separate, but they're now going to be different from one another, right? You see in the third picture, anaphase 1, of course, the polar microtubules are unsliding, so that's pushing the cell apart. But the ones that are connected to the chromosomes have pulled the pairs apart, not each individual one. Remember, they're not attached to each individual one. They're attached to the outside of the pairs. So it's going to separate the X's if you're looking at that picture. So it's going to pull them apart. If you're wondering, if you look at them, the little different color on the tips, that's the swapping of the genes. So that's when the physical part, right? That's what I mean by these stages blending into one another. The crossing over, the chiasmata were formed at prophase 1. They moved into metaphase 1. The spindle fibers finished forming. And then when they went to anaphase 1, then the actual physical swapping of the information occurred. So by the time you get to telophase 1, you've already got cells that are different, right? And of course, an animal cell is going to split into 2 cells at this particular point in cleavage furrowing, isn't it? Okay? So we've cut the number of chromosomes in half. At the very beginning, you started with 6. How many chromosomes in that cell right there, the top one? How many chromosomes do we see? Not talking about humans. Look at that cell right there. How many chromosomes do we see in that one cell? Cleavage furrowing is finished. We now have 2 cells. So, right here then. How many chromosomes do you see in that cell? Same number you see in this cell. Chromosomes. I see. Come on. I count the X's, count the centromeres. It might help. 3 in each, right? How many chromatids in each? All right, there's 6 chromatids. No, there's chromatids because there's things duplicated, right? Don't worry. Assess question is a good one about, their information, but I do think it has more to do with the attachment. There you go. All right, so there's prophase 2. All right, so when we move from telophase 2, go back, or I'm sorry, excuse me, telophase 1, we separate the cells in half, and we've finished dividing the chromosomes. Now, what happens in purple, that's the nuclear envelope reforms. You remember, that's another thing that happens in mitosis. I don't think I mentioned it in prophase 1. I'm sorry if I didn't. But the nuclear envelope disappears. You got to do karyokinesis, right? The movement of the nuclear material, so you got to get that envelope out of the way. So in telophase 1, the envelope's going to start forming again. The spindle fibers go away. You don't need them anymore, right? They're just used for movement. So we're going to look like this when we move into prophase 2. Now, what kicks off prophase 2 is going to be the nuclear envelope starts to disappear. Why does it form and disappear? That's an important package. You're going to want to put an envelope around it anytime you're going to be splitting the cell around. You don't want pieces of the nuclear material squeezing out as the cell split. So package that off separately. But the spindle fibers are going to form. And again, if you're an animal cell, centrosomes and all that other stuff is going to happen. No crossing over, no synapsis, right? This is why meiosis 2 is sometimes called mitotic division. Don't get confused, but you're following 2 cells. But look at each one. This is just mitosis, right? You're not going to have a cell with just 3 chromosomes, obviously. But everything lines up in the middle in metaphase 2 with its swapped over parts. Everything separates. Now here, you do have kinetochore microtubules going to each chromatid. So the chromatids pull apart, sister chromatids separate, becoming daughter chromosomes in daughter cells, all genetically different. The picture is too small, but if you look at those 4 cells right there, you'll see that if you look at if you let them separate, and you look at the chromosomes, you got a short red one, then you got a red one with a little blue tip, and so on and so forth. But you don't need to do it, but these are all different. This is different, this is different, this is different, and this is different. And every time you make them, it's different. And where they cross over, it's different. And oh, by the way, not only that, when they line up like this, that's different. Do they line up with, the blue with the red with the little blue tip on the left, or the blue with the little red tip on the right? And when they line up here, they do it differently. And even in mitosis, when they line up, is it always going to be the maternal on the left and the paternal on the right? Now, I'll show you a slide of that in just a second, but randomness, folks. So we're making very random cells here. Okay, 2 more slides. First one I'm going to show you is the visual. Second one is the written. I'm not going to go through the second one. I'm going to show it to you because it's a good place for you to help get your ideas in order. This slide is 2 because it's mitosis versus meiosis. I want you to look at these next 2 slides on your own and convince yourself that you understand the terminology and how these things are the same and how they're different. How's everybody else hearing me? All right, so chromosome number and difference in chromosomes, okay? See, if you look at the meiosis cells down there below on the right, each one's different, isn't it? Each one in terms of what color variants you see are different. The one on the left, the cells are identical. Chromosome number stays the same in mitosis. It doesn't in meiosis. This is all stated right here. All right, so read through this. Don't worry so much about where it says gametophyte, sporophyte, but read through this. It's going to help you understand and keep it in your head, you know, how these things are different. Don't confuse these two. And, of course, the biggest thing to remember, number, right? If there's a number, then we're talking about a stage in meiosis. All right, I only got a few minutes, so do let me get into this, and then we'll stop and pick up the rest of it in class. But let's talk about where this genetic variation that we've been talking about comes from. As I mentioned, mutations do occur. Human beings, we know kind of at what rate mutations occur. It's called a molecular clock. But we know that every day humans create about 65,000 genetic mutations. In other words, changing that genetic code. Now, most of those in humans aren't even felt. They're what we call silent mutations for reasons I'll talk about when I get into genetics. Not all of your DNA is actually necessary or even read. I mentioned to you the way these things line up in metaphase, and then, of course, crossing over, and then the whole idea of random fertilization. So mutations, look at this. I've just taken a chromosome here, right? It's got 6, or I've taken a cell, excuse me, and it's got 6 chromosomes, and I've lined them up differently. And that's going to be variation because these are going to end up in different cells, right? So the one in the upper left, all the blue ones, the paternal ones will end up in one cell, and all the maternal ones will end up in the other cell. And then the one just to the lower right, it's the other way around. And then there's all these other ones, right? So this one up here in the upper right, one cell gets a blue, a red, and a blue. The other cell gets a red, a blue, and a red. And then, of course, they didn't draw it in here because they didn't want to confuse the picture too much. But these are all going to cross over and mix up. So there, if you want to know how many possible kinds of gametes, sperm or egg, you can make, you take how many people need to produce cells, 2, our case, a male and female, and the haploid number, which our case is 23, and it's over 8 million. You can produce 8 million different gametes, and the other sex partner, the male or the female, can produce another 8 billion. So to figure out the total, it's 8 times 8. Okay, I'll come back to this probably the next time. There's that 70 trillion. So then random fertilization. This will be the last thing I talk about. And I have 2 more slides, but I can go ahead and do this here. What sperm gets to the egg is all randomly figured out, right? The randomness of meiosis, then the sperm are produced. Gosh, no, I want to talk about more of this. I'm going to stop right there because I do want to talk about random fertilization a little bit more. So what we'll do, this will be the last slide right here. The fact that in metaphase, this is, you know, true in meiosis, obviously, with the crossing over. This is going to make all different cells. I'll talk more about random fertilization with variation later on. I don't want to rush you that. Alrighty, folks, that's it for today. If you have any questions, stick around. If not, have a good weekend. Got a couple of weeks where we don't have anything to do. Like I said, get working on those homework assignments, and you can start studying for the 3rd exam now. That'd be a good idea. Please don't ask me when it is because it's on the syllabus, so I don't have to. From here, this will be this slide, then we'll go from here. Yeah, get the extra credit out at Donald's Farm. I didn't get to go last weekend, but I'm going to try and get to go out there this weekend. This is the last weekend. 10 points. I'm going to turn the recording off, folks.